Dear student, today we will be discussing skeletal muscle system. Now, skeletal muscle system consists of skeletal muscles, and skeletal muscle is a form of striated muscle tissue which is under voluntary control of nervous system. They are attached to bone by bundles of collagen fibers known as tendons. They help in the movement and locomotion. So that is what is the main function performed by the skeletal muscles. Now, what are the special properties of these skeletal muscles? They are electrical excitability, extensibility and contractility. Electric excitability means they are capable of being activated by receiving stimuli. Extensibility means ability to be stretched. Contractility means they come back to their original position after being stretched. Now let's look at the organization of these muscles. The basic unit is myofibril. It is cylindrical, unbranched and elongated structure made up of protein, and this is called unit of muscle cell. Muscle fiber or muscle cell, they consist of bundles of myofibril and arranged parallel to each other, and they consist of all cell organelles. You can say endoplasmic reticulum, which is called sarcoplasmic reticulum, sarcosoma, which is mitochondria, sarcolemma, which is plasma membrane, sarcoplasm, which is cytoplasm, nucleus. Actually there are multi nucleus which are present, and this condition of muscle is called syntidial, and these nucleus are located at the periphery. Now the muscle fiber forms the bundle called fascicle. The bundle of fascicle form muscle. Now you look at the organization in order. So myofibril is there. Myofibril group together to form muscle fiber. Muscle fiber group together to form fascicle bundles, and they group together to form muscle. So muscle fiber are surrounded by endomysium, fascicle surrounded by perimysium, and muscle surrounded by epimysium. If you look at composition, myofibril consists of proteins. Muscle fiber has all the cell organelles. Fascicles are supplied with blood capillaries and nerves, and muscles, it is attached to the bone via tendon. Now look at the diagram. So here I'll take another order to explain you the diagram. This is the bone and this is the muscle, the purple one. And the muscle is attached to the bone by tendon. You can say the muscle is covered by epimysium. This is the layer of connective tissue. And then you can say it is made up of so many fascicles. Okay. Now if you look at the structure of the fascicle in turn consists of so many muscle cells. Okay. And the fascicles are surrounded by perimysium. They are supplied with nerves and blood capillaries. And if you look at the fascicle consists of muscle fiber or muscle cell. Okay. So muscle cell has all the things. The nuclei are located in the periphery. Then it has sarcoplasmic reticulum. This is cells surrounded by plasma membrane, which is called sarcolemma, and which in turn is surrounded by endomysium. Then you can see each muscle fiber has a number of bundles of myofibrils, and these myofibrils are myofilaments. They are also known as, and made up of proteins. We'll see what all proteins are present. So here now first I would like to discuss the structure of myofibril. So if you look at myofibril, they appear striated due to the presence of light and dark bands which are alternately present throughout the length. So if we see them under the microscope, you will find two types of band. Light band called I band or isotropic band, dark band called A band or anisotropic band. The light band, why they are known as, because light can pass through them, so they appear lights, and through dark the light cannot pass through. Composition, if you see, a light band is made up of actin filaments and Dark band made up of myosin filament. If you see in the center of light band where a line bisect, this line is called Z line. Similarly, at the center, thick H line is present, and again in the center of H line there is M line. So you just look at the organization. The green highlighted portion represent dark band, the orange highlighted represent the light band. So you can see alternate light and dark band. So if you see in the center of light band there is a line, this line is called Z line, and in the center of dark H line is present, and then there is M line which is in the center of H line. So this is how it organizes itself. Now if you see the distance between two Z line, this distance is called sarcomere, and sarcomere is said to be the structural and functional unit of myofibril, and it consists of half I band, one full A band and half I band. Each myofibril consists of actin and myosin. Okay, now we will see the working of muscle. Now how the muscle work. So basically how they function by simultaneous contraction and relaxation. Now what are the requirements of these muscles? Proteins, energy, calcium, oxygen. Protein are of two types, contractile and regulatory. Contractile protein are those protein which help in the contraction, and regulatory protein actually regulate the contraction process. So contractile protein are actin and myosin. Regulatory protein are tropomyosin and troponin. Now energy is obtained from ATP. ATP is synthesized by the breakdown of glucose. Now glucose is obtained from the glycogen in the muscle cell, since glycogen granules are present in the muscle cell. Calcium plays role in the signaling, we will see later how. And oxygen is required for ATP formation, respiration process where glucose breakdown and releases the energy, and this ATP formation occurs in the mitochondria. Myoglobin is an oxygen binding protein which is present in the muscles, and spatially it acts as a carrier for oxygen just like hemoglobin in the blood. Now let's discuss one by one the protein. Actin protein, which is present in the I band, is a globular protein. Its monomer form is called G-actin, and its polymer or the filamentous form is called F-actin. So actin is made up of two helical of F-actin. So if you look at the diagram, there are two helix. Okay, one circle of this, you can say one circle is called G-actin, and it polymerizes and form this chain which is called F-actin. So two chains of F-actin are arranged in the helical manner. Now look at the structure of myosin. It is filamentous protein, and its unit is called meromyosin and has a tadpole-like appearance. So if you see what meromyosin consists of, two heavy and light chain. Now these are, because their molecular weight differs, depending on that they are known as heavy and light chain. If you look at the structure of let's first discuss the heavy chain. So four heavy light chains are there, and they further consist of two essential light chain and two regulatory light chains. The two heavy chain consist of tail and head structure. Tail is the rod shaped structure, head is a globular structure, and the head also consists of four light chain. So head has two heavy chain plus four light chain. Okay, so head consists of so many other things. Function wise, it consists of ATPase activity. ATPase is the enzyme that catalyzes breakdown of ATP to ADP and releases the energy. It also has a binding site for actin, and head is involved in the cross bridge formation with active site on actin filament. Okay, so first look at the diagram, meromyosin and myosin. You can say this is the head part, globular, and tail part. These are four light chains, you can say, see. Okay, and you can see the binding site for actin, which is located on the head of meromycin. So this is a unit and it polymerizes and give rise to myosin. So the myosin protein look like this. Okay, the heads are projecting out, and these heads has the two active ATPase activity, plus they bind to the actin part. Now also look at the tropomyosin. It is rod-shaped fibrous protein. The rod-shaped structure join end to end and form two helical strands which wrap around actin. It covers the myosin binding site on actin. So this is how the tropomyosin look like. Now troponin consists of three subunits. They are present at regular interval on tropomyosin. They mask the active site on F-actin during resting phase. So if you look at the complex, F-actin form the complex with the troponin and tropomyosin. So blue color is, you can say, F-actin, then tropomyosin, okay, and then troponin. So tropomyosin and troponin together, what they are doing, they are masking the active site. There is some active site which is present, and this active site is required by the myosin to bind over here. Okay, so this complex is called tropomyosin troponin actin complex. Now let's look at the muscle at rest and position of actin and myosin. So again come back to the dark and light band, how they are located and where these proteins are located. So this complex is located in I band, whereas the meromycin located in the dark band. During the resting phase, the actin and myosin filament lie parallel to each other, in which actin partially, you can see, overlap at the two ends of myosin. So dark and light band, you can see orange and green, whereas the blue line over here represent the complex and the orange line represent the myosin structure. So partial overlapping here, you can see this is A band and I band. Then during contraction, the active filament slide toward H line till H zone disappear completely. The I band width decreases. The A band width remain constant during relaxation and contraction both. Now over here you just look at, see, I band disappear. Why? Because they slide toward the dark band, and you can see this is the sliding mechanism that has occurred. So if you look at the sliding mechanism, which is given by Andrew F. Huxley and Rolf Niederjerke, the actin filament slide over myosin toward inward, body side toward the inward. Head of the myosin operate like hook and attaches to F-actin, forming cross bridges, thus pulling the actin filament toward inside. The sliding mechanism require energy that is obtained by the ATP through ATPase activity of myosin head. Formation of ectomyosin complex occur. So when the myosin bind toward the actin, now how the binding occurs, I will just produce the recap. The myosin head has the ATPase activity, and this also has the active site to bind at the you can say F-actin. So when it bind, the energy is required for the binding, and myosin head only releases this energy because it has the enzyme ATPase. ATP is released, and the head of the myosin act like a hook which slide on the actin filament. So overall there is decrease in the length of sarcomere, you can see, okay, due to shortening of I band, where the length of A band remains the same. The H zone disappeared completely, and here you can see this is ectomyosin complex, the head, okay, and here you can see this is F-actin complex, myosin, they bind with each other and head look like a hook, and this complex formation takes place. So next part we will continue in part 2. Thank you.